the promise of gene therapy and how we catch illnesses early. Hey future explorers! Welcome back to the channel. Have you ever wondered if we could fix diseases that are passed down through families? Diseases we are born with? Well, today, we are diving into some mind-blowing science that's attempting to do just that, gene therapy. And we'll also explore how scientists are getting super good at catching diseases way before they even make us feel sick. Imagine a tiny typo in your body's instruction manual, your DNA. If that typo is in a crucial gene, it can lead to a hereditary disease. For the longest time, we could only manage symptoms. But what if we could correct that typo? That's the dream of gene therapy. It's a collection of incredible methods that aim to correct a gene defect diagnosed in a child or even an embryo. Essentially, we are inserting healthy genes into a person's cells and tissues to treat a disease. The idea is to deliver a normal, working gene into the individual or embryo. This new gene then takes over the job of the non-functional one, essentially compensating for the missing or broken instructions. And guess what? This isn't just sci-fi anymore. The first clinical gene therapy was administered as far back as 1990. To a 4-year-old girl named Ashi De Silva, who had a condition called adenosine deaminase, or ADA, deficiency. Now, ADA deficiency is a big deal because this enzyme, ADA, is crucial for our immune system to function properly. Without it, the body struggles to fight off infections. This disorder is caused by the deletion of the gene responsible for making ADA. Before gene therapy, options were limited. Some children could get bone marrow transplants or enzyme replacement therapy, where they were given functional ADA by injection. But here's the catch with both of these: they weren't completely curative. They managed, but didn't truly fix the underlying problem. So, how did they approach it with gene therapy for ADA deficiency? As a first step, they took lymphocytes, a type of white blood cell from the patient's blood, and grew them in a lab dish. Then, they introduced a functional ADA gene into these lymphocytes using something called a retroviral vector, basically, a modified virus that acts as a delivery vehicle. Once these cells had the new gene, they were returned to the patient. Pretty clever, right? However, there was a challenge. These lymphocytes aren't immortal, so the patient needed periodic infusions of these genetically engineered cells. It was a groundbreaking step, but not a one-and-done cure. But imagine this: if that healthy ADA gene could be introduced into the cells at a very early embryonic stage, essentially, at the very beginning of life, that could lead to a permanent, lifelong cure. That's the ultimate goal in many cases of gene therapy. Okay, so gene therapy is about correcting the problem. But what if we could detect these problems, or any disease for that matter, super early? Because let's be real, for effective treatment, early diagnosis and understanding how a disease works are incredibly important. Conventional methods like analyzing your blood or urine are great, but they often can't catch diseases in their earliest stages. This is where some truly revolutionary techniques come in: recombinant DNA technology, polymerase chain reaction or PCR, and enzyme-linked immunosorbent assay ELISA. These are the superheroes of early diagnosis. Normally, you only suspect a pathogen like bacteria or a virus when you start feeling sick, right? By then, the pathogen's concentration in your body is already quite high. But what if we could detect even super low concentrations of bacteria or viruses, even before symptoms appear? PCR makes this possible. PCR is like a microscopic photocopier for DNA. It can amplify tiny amounts of nucleic acid, that's DNA or RNA, until there's enough to detect. So, even a handful of viral particles can be multiplied millions of times, making them detectable. This is why PCR is routinely used to detect HIV in suspected AIDS patients or to identify mutations in genes in suspected cancer patients. It's a powerhouse for spotting many other genetic disorders, too. Think about it: a single-stranded DNA or RNA, tagged with a radioactive molecule, we call this a probe, is allowed to bind to its complementary DNA in a sample of cells. Then, using autoradiography, we can see if it is bound. If there's a mutated gene, the probe won't find its perfect match, and that area won't show up on the photographic film. It's a brilliant way to pinpoint those tiny genetic changes that can cause big problems. And then there's ELISA, based on the principle of antigen-antibody interaction. Our immune system produces antibodies to fight off invaders, called antigens. So, an infection can be detected either by directly finding the antigens from the pathogen itself, or by detecting the antibodies your body has made specifically against that pathogen. It's like finding either the criminal or the police force that's been specifically trained to catch them. So, from the revolutionary potential of gene therapy to rewrite our genetic code, to the incredible precision of PCR and ELISA in catching diseases at their earliest whispers, science is constantly pushing the boundaries of what's possible in medicine. We are not just treating symptoms anymore, we are aiming for cures, and we are getting smarter about preventing and intervening before things get serious. What do you think about these incredible advancements? Let me know in the comments below. If you found this video fascinating, smash that like button, subscribe for more science awesomeness, and hit the notification bell so you don't miss out. Thanks for watching, and I'll see you in the next video.